All right. Thank you so much for the invitation to speak at the seminar, which I always enjoy. So today I'm going to be talking about HIV dynamics, HIV infection, and how mathematical models can help us understand the dynamics of HIV and how it evolves, really the evolution of the virus. So if you have any questions at any point, don't hesitate to jump in and ask. I'm happy to be interrupted. So the talk, I'll start out with an introduction. I don't know the composition of the audience. I'm going to give a very brief introduction to viruses, immune system, HIV, then talk about mathematical models of virus dynamics and HIV dynamics. Just simple models that are sort of the bread and butter in the field, and then I'll move on to compartmental structures, because a lot of models do not take into account the structure of lymph nodes. At the same time, lymph nodes are the primary site where HIV replicates in patients. So it's kind of important to do so. And so I'm going to explore the effect of the compartmental structure on viral evolution, in particular immune escape evolution, and also general evolutionary dynamics. And I would like to point out that this work is kind of freshly finished / still in progress. So I mean, some things are better worked out, and maybe towards the end they're less worked out. But nevertheless, I like talking about it because it's new stuff and interesting. And all this work was done by my graduate student Wen. He's now just started a postdoc at Guelph. And we did this in collaboration with a couple of experimental people, Elizabeth Connick at Arizona and Pam Skinner at Minnesota, who are doing experiments with SIV-infected macaques that are relevant to this theory. So let's get going with some basics. We're going to talk about virus dynamics. What is a virus, and what kind of processes do the mathematical models describe? A virus has to go in a cell in order to replicate. It can't replicate outside a cell. So the basic process that we're modeling is that you have a susceptible cell that is uninfected. You've got a virus. The virus goes into the cell. The infected cell produces new virus particle, releases those, and you make more infected cells, and the virus population amplifies in this way. So it's just very basic stuff. I'm going to be talking about immune cells, and I'm going to particularly talk about CTL, which are called cytotoxic T lymphocytes. And so here's an overview of the adaptive immune system, which is very instrumental in fighting viruses. There are basically two branches: the B cells, which produce antibodies. I'm sure everybody has heard about those in the context of vaccines, but they're also the CD8 T cells, or CTL, cytotoxic T lymphocyte, or killer T cells, and they kill infected cells. And they're particularly important for viral infections because cells are infected, and to remove them, you got to kill them. And so this branch of the immune system is often very important, and is especially important in HIV infection. So that, just putting into context the CTL, this is what we're going to be talking about. This talk is about HIV. HIV is a retrovirus. This means so its genome consists of 2 RNA copies. It's diploid, surrounded by a protein coat. And it's a retrovirus because the RNA, when it enters the cell, is copied into DNA. It integrates into the genome of the host cell and becomes part of our own genome upon infection. So this is sort of an outline of the virus that we're talking about. HIV has a certain profile in terms of its natural history of infection. The best marker to look at in this graph is kind of a stylized graph, is the virus shown in yellow. There are 3 phases to the infection. The first phase is the acute phase of the infection. That's just when the patient gets infected, and this happens for like a month or 2 or 3. That period, the virus grows massively to very high levels. Patients may experience symptoms typical of any virus infection, non-specific symptoms. And then immune responses kick in, and the amount of virus is reduced to low levels, and that's where the chronic phase or the asymptomatic phase starts. Patients do not show any symptoms. They typically have very low virus load, and they appear completely healthy. And this can last for varying amounts of time. It can, on average, it's like lasts for 5 to 10 years. There are some patients who stay asymptomatic for more than 20 years or maybe forever. We don't know. These are elite controllers. There are some patients that progress pretty fast, but on average, after about 5 to 10 years, the virus starts to expand again, to grow, and this correlates with the onset of symptoms, immune deficiency, which is called AIDS, acquired immune deficiency syndrome, and eventually, if untreated, this leads to death. And the other important measure is the blue line, the CD4 cell count is around 1,000 per milliliters cubed per milliliter, and it goes down upon initial infection and then gradually goes down until when AIDS develops, it's at a critically low level, such that immune responses can't be mounted anymore. And that's where AIDS sets in. And so this whole process, I mean, weirdly enough, there's a lot of progress in treatment, but this whole process of what drives disease progression is actually not that well understood. But we do know that viral evolution plays a critical role in pushing this disease from the chronic asymptomatic phase to the AIDS phase. And so studying viral evolution, understanding how the virus evolves and the timing of viral evolution is important in that respect. And that's what motivates all these studies, which is also what I'm talking about now. So how do we model virus dynamics or infection dynamics? It's very similar to how we model infections on an epidemiological level. Everybody heard of COVID modeling, I'm sure. And this is kind of the same thing. In epidemiological modeling, you have susceptible, infected, and sometimes recovered. Here you have different kinds of compartment, which is uninfected cells, infected cells, and free virus. And this is the, whoops, 1 second, there's something, I don't know why it's doing, whoops, why it's doing that. This is the model in schematic form. Basically, you have the uninfected cells. They are produced at a given rate lambda. They die at a rate d in this notation. When they meet a virus, there's infection happening with the rate beta. The infected cells then die with a rate a, and viruses produce at a rate k and decays at a rate u. That's sort of the basic model of virus dynamics, which underlies a huge number of studies that have been published over many years. And so I'm just introducing that because what we are going to do is also based on that. So I'm going to be talking about more complicated versions of this. So this basic model has two equilibrium outcomes. One is the infection-free equilibrium. The other outcome is an equilibrium where the infection persists. This is a simple model, doesn't have immune responses, but gives you some basic properties. An important property of this model is the basic reproductive rate or the basic reproductive ratio of the virus, which is the average number of newly infected cells generated by a single infected cell at the beginning of the infection. And so if R0 is bigger than 1, the infection spreads and becomes established. If the R0 is less than 1, the infection goes extinct. Again, similar to the epidemiological modeling that we have heard quite a bit about in recent years. So that's just introducing those concepts. So if you want to expand this model, the next level of complexity is that you include immune responses in this model, such as CTL response. And this is how you model a CTL response. Denote this by Z here. The CTL response expands at a given rate, which you call C, when it sees infected cells, Y. And in the absence of stimulation, the CTL response decays. And when the CTL are around, they kill the infected cells with the rate P here. And these things can be formulated in different ways. These are highly phenomenological models where we just, on a very rough scale, model the expansion of immune responses. And so that has to be kept in mind. But this is generally how it looks like. And then you can run these, simulate these. You can get either persistent infection where they kind of go to a stable equilibrium, or you can essentially effectively eradicate a virus if the immune response is strong enough. In an ODE, it will only go to very low levels, but corresponds to eradication, basically. So it can mimic what happens in real life in immune responses. And this formulation, it's similar to predator-prey dynamics, where the CTL act kind of like the predator and the infected cells act like the prey, and that's why you also can see these sort of oscillatory dynamics that happen. So that's CTL responses. So now we're going to be talking about evolution. And in particular, one thing we're talking about is the escape of from CTL responses, the viral escape from CTL responses. So viruses such as HIV evolve. They can easily generate mutations, point mutations, and when the virus has a mutation in the part of the virus that is recognized by the immune response, that is recognized by the CTL, then it is not recognized by the CTL anymore. And this is what we call an escape mutant. The virus can then grow without being opposed by the CTL and can cause more trouble. And this is just a way of how this is incorporated into these kinds of models. Now we just don't have one type of infected cell and one type of virus, but we have two types of infected cells and two types of virus. The Y1 is the cells infected with the wild type. The Y2 is cells infected with the escape mutant, and the corresponding free virus populations are the same. And you can see that the killing is. Can you actually see my mouse? Yes. Okay.
Yeah, we can see it. Yeah.
Okay, that's good because I wasn't sure. So the CTL kill the cells infected with a wild type, but a corresponding killing term is not included for the mutants, and so the mutants can escape this suppression and grow to higher levels. That's sort of the basic structure of what we're talking about.
Dominik, just a quick question. In the first equation for X, I assume that the last term is a V2 and not
V2,
a V.
yeah.
Okay, sorry, just making sure.
Yeah.
That's okay.
Didn't see that. Yeah, good. Okay. Yes. Okay. So this kind of model has been used in a variety of studies. Immune escape has been studied in a variety of settings. This is a multi-strain immune escape where immune escape pops up and maybe other immune responses come and recognize the escape mutants, and it's like a cat and mouse game where the virus always escapes immune responses and the new mutant is then recognized, eventually breaking free. That's just one of the things that has been done, but there's been a lot of theoretical study of immune escape and the principles of evolution of immune escape, how long it takes, and how likely it is, and so on. So a big literature exists on that. And so now I want to come to where I think some of the limitations are, and this is how HIV is measured and how these and consequently how all these models are constructed. So when patients come into the clinic, and the doctors will assess their state by drawing blood and measuring the amount of virus in the blood. And you can also measure the immune responses. You can measure the number of CTL directed against specific parts of the virus. And you can follow that over time. And you can fit models to it. Examples are shown here, the types that I just went over, and this gives good fits, and you can get various conclusions from that. And so the models, as I shown so far, they're all ODE based models, or not all of them, but a lot of them. This is a major means of studying virus dynamics, which means that we assume that viruses and cells mix perfectly throughout the system. And this is where reality is more complex. And the first thing to point out is, although we're measuring the virus in the blood, there's actually, and blood cells are infected, there's actually no virus replication that goes on in the blood. Where the replication happens is in the lymph nodes in the spleen, the lymphoid tissue. That's obviously where also a lot of immune cells are located, but it all happens in there, and the blood is sort of just an image that you get where things get obviously flushed out from the lymph nodes into the blood, and so you can get some sort of a picture of what's going on. But where things really happen is in the lymphoid tissue, and the lymphoid tissue has a very complex structure, and this is actually a simplified diagram here. And what I want to focus on in these lymph node structure is that there are two types of compartment. One is the follicle, which is called follicular compartments, B-cell follicle, and then there is the area outside the follicles, which is the extra-follicular compartment. And so I abbreviate this usually extrafollicular with an EF and follicular with an F, because these words are kind of a mouthful. So I refer to them as EF and F. And there are important differences in these between these two compartments, and especially when it comes to the CTL response. So the CTL response is primarily generated in the extrafollicular, the EF compartment, and it can be very powerful in there. However, you do not really want the CTL to enter the follicles, the B-cell follicles, because they would cause trouble there, they would wreak havoc there. And so this would be a bad strategy. So the immune system is designed in a way that when immune responses arise, they get generated in the extrafollicular compartment. And for most purposes, like in lymph nodes for regular infections, this is just fine. HIV happens to infect the lymph nodes, happens to infect the immune cells, and so that's where it gets tricky, because HIV can go and infect a lot of cells in the follicle, and so it has an immune privileged sites in the follicle. So let me show that in a more a sort of caricature kind of way. So here you've got the extrafollicular compartment and the follicular compartment, and I depict what I think are sort of important processes that are going on. So the first thing to point out is that these are two separate compartments, and so you have compartmental structure going on. So the CTL, they get stimulated by the virus in the extrafollicular compartment, and they will expand here, and they can very efficiently fight this virus population, which is these red things here, and get rid of them. So every now and then, some CTL will enter the follicular compartment, but they don't really belong there. So this migration rate, we can say in a biologically realistic setting, this migration rate is very slow. It just doesn't really go there a whole lot. And once a CTL has made it into the follicular compartment, there's a huge migration rate back. They home back to the extrafollicular compartment. They need to get the hell out of there, so they don't kill things they're not supposed to be killing. They don't cause inflammatory processes in there. So they're not wanted there.
A quick question here.
Yeah.
Is it clear why the CTL migrate at all?
Why they migrate at all?
Yeah.
Oh,
Is there any mechanism why they migrate? Is it fusion? What is it?
It's a consequence of the fact that cells can move from one compartment to the other, but they home to certain parts, and so they're more likely to stay in the extrafollicular compartment. But there's nothing like not a barrier as such preventing them from going there. Does that make sense?
That's why it's a very low rate.
Yeah, that's why it's a very low rate. You can engineer the CTL such that they will home to the follicular compartment, and that's actually in HIV one of the one approach they're looking for in terms of immune treatments, whether you can enhance that, although it's unclear what the side effects of that would be. But that's an engineered CTL where you modify the homing properties of the cell, but naturally they home to the extrafollicular compartment, and if they make it across, they go back rather fast. So this going back is always is fast. This going there is slow. So this is compartmentalizes the CTL such that most of the CTL are located in the EF, in the extrafollicular compartment, and almost none of them is located in the follicular compartment. There's also an exchange of infected cells or viruses. They can go across. This is thought to happen at a low rate as well, and happens mostly at the boundary where these two compartments meet, where they can just kind of go across. So we can assume that these are low rates in biologically realistic settings. So what I really want to point out is I've taken the CTL out of the follicular compartment now, because although a few can make it there, practically this is not very important. So this is what we can call an immune privileged site, where the virus can actually grow rather uncontrolled, and there it's largely controlled in the extrafollicular compartment. So this creates an interesting scenario where there's unequal distribution of viruses that can arise, with a lot of virus in the follicular compartments and very little virus in the extrafollicular compartment, because they're controlled by the CTL. And so here's some data that documents this. And so we can look at, on the left first, this graph that is labeled B. This is data from SIV infected macaques. So they infect monkeys with SIV, which is a equivalent of HIV in primates, and then you can see how much is of the virus. You can take biopsies from the lymphoid tissue from the two compartments, and you can see how much virus there is in F in the follicular and EF in the extrafollicular compartment. And first look at this acute phase of the infection. This is, remember I talked about the early acute phase, later chronic phase. In the early acute phase, the CTL responses have not had time to rise yet. It takes time for them to rise. They're mostly there in the chronic phase of the infection. So we have essentially no CTL in the acute phase of the infection. You can see in the monkeys, the different lines are different animals, and they largely have an even distribution of virus across the two compartments. And the reason is there's no CTL around, and so there's nothing to cause an uneven distribution. But now let's look at the chronic phase of the infection, where CTL responses can be there. And different animals can vary in the strength of the CTL response. Some animals have very weak CTL responses and bad virus control. Others have stronger CTL responses and stronger virus control. And you can see here, the ones with the better CTL responses are the ones that have the low amount of virus in general, and that's sort of the lower points. And you can see a clear difference in the abundance, with significantly more virus being present in the follicular compartment and very few virus in the extrafollicular compartment. Now, if you look at those animals that don't control the virus well, these don't have very efficient CTL responses at all. There it's again more equal. And this data suggests that if there are reasonably strong CTL responses around, you will have an unequal distribution of virus, with most of the virus being in the immune privileged, in the CTL privileged site in the follicular compartment, and rather little virus being in the extrafollicular compartment. And this is also shown on the right here in this graph that is labeled as F. This is the ratio of F to EF on the x-axis here, is the log of that, and the amount of virus on the y-axis. And so you can see if you have a lot of virus up here, your ratio of F to EF is 1, or 10 to the 0. It's a weird notation, but it's 1. They're similarly distributed. But if there's little virus around, controlled by the CTL, then you have a F to EF ratio that's much bigger than 1, and means you have many more viruses in F than in EF. Same story, basically. So that's the data that we have, and these are the data that came out of Elizabeth Connick's lab, with whom we have been collaborating. And so we wanted to explore this in terms of models. So we're interested in evolution, but we started out by having just a model without evolution. And this is basically this simple model of virus infection that I explained to you earlier, formulated in two compartments. And here we don't have a free virus population. We only have uninfected cells and infected cells, because we assume that the virus is simply proportional to the number of infected cells. Virus turns over very fast compared to infected cells, so we can kind of assume they're in quasi-steady state and ignore them. And yeah, so you have the uninfected cells in the EF, in the extrafollicular compartment, the infected cells there, and the CTL, and you have the same population in the follicular compartments. And the important things are these migration rates of movement rates. With eta, we denote the movement of infected cells from one compartment to the other, so they can go from one to the other with equal rates, essentially. Then we have this low migration rate G of CTL going from the extrafollicular compartment to the follicular compartment, and they move back at a fast rate H. And you can see there's a difference in the CTL equation in the extrafollicular compartment compared to the follicular compartment. In the extrafollicular compartment, you have an expansion term. So here the CTL gets stimulated and they expand in here, but in the follicular compartment, this term doesn't exist because they have no business there. They don't expand in there. All they do is they can kind of at a slow rate migrate into there, and then they can die, and then they can move back to the extrafollicular compartment where they belong. So we've got this migration rate eta, which is the exchange rate of infected cells, and the migration rate G, which is the slow migration of CTL from the extrafollicular to the follicular compartments. And we're going to be playing with those rates and exploring how they influence things. And we don't really know what their parameter values are because they have not been measured in vivo, or they have not been. We don't have numbers. We don't have rates. So how does the system behave depending on these parameters? And so here we vary the migration rate of infected cells eta, and on the right hand side we vary the migration rate of CTL from extrafollicular to follicular compartment, which is the parameter G. And blue is the F compartment, the amount of infected cells in the F compartment, and red is the amount of infected cells in the EF, the extrafollicular compartment. So you can see in the extrafollicular compartment, the amount of infected cells is always low because the CTL are really powerful there, keep them at low levels. And as long as the migration rate is relatively low, both of them, there's a lot of virus in the follicular compartments. If you now increase the amount of mixing, which is probably less biologically realistic, but you increase it, then the amount of virus in F goes down due to the increased mixing. It's simply they migrate between compartments, and more cells, more virus in the F compartment gets attacked by the CTL, and you get lower and lower infected cells in the F compartment. So what is biologically realistic? We think that any of these migration rate regions where there's this big discrepancy in virus load between F and EF, that's where it's realistic. So this could be where this arrow is on the left or this arrow. We don't really know how low the migration rate is, but what we explore is parameter region where the virus load is clearly separate. And when they start kind of converging, this is where we know from the data that this doesn't happen. So this is the region of migration rates that one can consider realistic. So just some basic properties of this model. The model can basically recapitulate these experimental data from the SIV-infected macaques. This is a simulation and results of simulation, or where we plot the equilibrium number of infected cells against the strength of the immune response, which is the immune responsiveness with the parameter C. And just as the data depict, if the immune response is weak, the amount of virus at equilibrium in F and EF is similar, but as the immune response gets stronger, there's a big discrepancy emerging where there's little virus in EF and a lot of virus in F. So that recapitulates these experimental data, and we can also plot it in this form of the ratio of amount of virus in F to EF versus the total equilibrium number of infected cells. If the total equilibrium number of infected cells is high and immune response is basically not very effective, then the F to EF ratio is 1, just like in the experimental data, and it goes down if the virus load is lower because they're more CTL now that results in that compartmentalization. So that's good because we have a model that can recapitulate what the data shows us, and that's always comforting. So one thing we wanted to look at is some very basic dynamics. Before we get to evolution, we wanted to examine, can this compartmental model explain something in HIV infection that is hard to explain otherwise? And we think it can. And so what we looked at is antiviral therapy and the rate of viral decline during antiviral therapy. And this is a schematic that shows you what happens when you start treating an HIV-infected patient with drugs. And so the schematic down here shows you what happens when you apply antiviral therapy. So antiviral therapy does nothing to cells that are already infected, but it prevents the infection of new cells. And it prevents the spread of the virus to new cells. So that's what the drugs do. So as a consequence, you cut the transmission, the drugs cut the transmission, and then what happens is that the cells that are infected and that already exist, they start dying. And that's why you see a decline slope of the virus. And they can die because the virus kills the cell, and they can die because the CTL kills the cell. So these are the two forces that can potentially cause the death of the infected cell, and that should be reflected in the decline. And so you see a decline of the virus population, and this decline slows down eventually because this is a sort of latent reservoir largely where the virus is not active, so these cells don't die a whole lot. But what is sort of important is this first phase of decline. And this was a very important paper where they showed that the half-life of the infected cells is actually very short. They decline very, very fast. They live only like 1 to 3 days, essentially. So this is the slope of virus decline. And the slope of virus decline during treatment has gained a lot of attention. There's a lot of research into that. And the reason is that this is very, very constant across patients, always around the same values. And this is a bit intriguing. And so this is shown here. This is the half-life of infected cells during treatment. So this decline slope here, plotted against CD4 cell count. And CD4 cell count is, that's the target cells of HIV. And if there are lots of CD4 cells still around, then you have pretty much strong immune responses still, strong CTL. If you've got very few CD4 cells around, then your immune system is largely gone and the patient has AIDS, and there's probably no CTL that are there. Nevertheless, this decline rate, the half-life of infected cells during therapy is the same, regardless of the CD4 cell count. So you see this incredible constancy. And this doesn't make sense on some level, because the CTL are likely to contribute to the death of the infected cells. So to this decline here, you have CTL that are around, they should be hammering the infected cells here. And so the question is, why is this rate of decline the same, whether the CTL response is already completely weakened or whether the CTL response is rather strong? You can make that point even stronger. You can look at the rate of decline during treatment in SIV-infected monkeys, and you can compare a normal animal with an animal where the CTL or CD8 cells are depleted. You can deplete them by generating antibodies to the CTL, and so you don't have any CTL anymore. And you can see that there's essentially no difference in the decline rate, whether they're CTL or not. So why is that? If the CTL are killing the infected cells at significant rates, which we know they do, why is it that you have the same rate of decline if there are no CTL that kill cells here, or if there are CTL that kill the cells here? So in principle, you would expect a difference in the slopes of decline. When the CTL are there should be a steeper slope of decline because the killing rate is faster. And so this kind of stuff has generated a lot of debate in the field. And some of the conclusions that have been reached is that maybe these CTL don't really do as much as we thought they do. Maybe they don't kill. Maybe they suppress the virus in different ways, because obviously if they were killing, you would see a difference in slope. So can we get information from that from our compartmental model? And indeed we can, because of the compartmentalization, we think is that you don't see a difference between CTL depleted and animals that have CTL. And so we made simulations. The solid line is a simulation with CTL present. The dashed line is a simulation with CTL depleted. And this is the dynamics that occur in the EF compartment, in the F compartment, and then the sum of the two, which is most likely to be in blood, which is what is being measured in clinical data. So in the EF compartment is, you see what makes sense. There's CTL present, and they're killing the infected cells. So initially, the rate of decline of the infected cells is faster if the CTL are there, the solid line, compared to if the CTL are not there. So there you see a very clear difference, which makes sense if CTL do a lot of killing and control the virus. However, in the F compartment, the follicular compartment, the CTL don't enter. So the decline rates, whether CTL are there or not, are only driven by virus-induced death, and they have identical decline rates in the F compartment, simply because the CTL don't really make it there. And so if you sum them both up, you basically get what is in the F compartment, and this masks what is going on in the EF compartment, because the virus is so much more abundant in the follicular compartments compared to the extrafollicular compartment. So if you sum them up, you will see no difference in the rate of decline of whether CTL are there or not, just as you see in clinical data. But this happens despite our assumption that the CTL do a lot of killing and contribute to virus control, but they do so mostly in the extrafollicular compartment. It's just that this is because CTL are so strong here, virus load is low, and therefore any differences that you would see in the EF compartment are masked by this bigger amount of virus in the follicular compartments where the CTL don't enter. So we think that if you do take into account the spatial structure of the lymphoid tissue and the processes that go, the compartmental processes that go on there, then you can explain why you don't see a difference in the decline rate during treatment in the presence or absence of CTL. And so we think that's an important point to make. So let's move on to evolution of escape mutant, because there's interesting stuff that just happens here, just if you look at the at this whole structure. So how do CTL escape mutants evolve? They get generated by point mutations, essentially. The mutation rate is around the order of 2 × 10⁻⁵ per base per generation, which is a high mutation rate compared to what happens in humans. In both compartments, mutants can be generated, but there are important differences. If you look at the follicular compartment, where there are hardly any CTL, you've got high virus load going on. Because you've got a lot of virus in here, it is very likely, it's much more likely to generate a mutant in here, in the follicular compartment. However, there is no selection pressure in the follicular compartment. The advantage of the mutant that is generated is minuscule, because it escapes a CTL response, but there is no CTL here. In fact, in the absence of CTL, this might even have a disadvantage, this mutant. And so it's very hard for it to grow in the follicular compartment. On the other hand, in the extrafollicular compartments, you have huge selection pressure, because you have the CTL that constantly kill the cells. So if you've got an escape mutant, it has an instant advantage and rises to high levels. On the other hand, there's very little virus in here, because the CTL kill it. And if there's very little virus here, the probability to generate a mutant is low. So you've got kind of interesting things that are going on here, and we wanted to explore what happens. And so we take a model. This is the model. I'm not going to go through the whole thing. It's kind of expanding from the previous model, where you have dynamics in extrafollicular compartment. E and F is the follicular compartments. Just see I have a mistake here. This is supposed to read F, but it doesn't matter. It's the same model as I showed you before, but we added CTL escape, so you have a mutant that is generated and that is not killed by the CTL response, and this mutant can be in both compartments. And so we want to study the time it takes for a mutant to invade essentially the extrafollicular compartment, because it'll never invade the follicular compartment because there's no selection pressure there. So we want to look at the time until the mutant virus in the extrafollicular compartment becomes dominant. And to do so is we run stochastic simulations of this system by using a Gillespie type algorithm. We start with a wild type virus at equilibrium as predicted by the ODEs, and their mutations being generated at a given rate mu. The mutants are generated, and so we let it generate, and we determine the average time it takes for the mutant to reach 95% of the virus population in the EF compartment. And the reason we don't go to full fixation is because it never fully fixates, because there's always the wild type that is in the follicular compartment that can migrate into there, and so they will always coexist, but they can become dominant, they can rise to dominance, and so we do 95%. And this is what I'm showing here, just sort of the outcomes that you can observe. Left is extrafollicular compartment, right is follicular compartment, strong CTR response on the top, weak CTR response on the bottom. You can say that with a strong CTR response in the EF compartment, the mutant, which is in dashed line, will come up and dominate. In the F compartment, it will never do so because it doesn't have an advantage there because there's no CTL. If there's a weak CTR response, it will not dominate in either compartment. So that's the rationale for doing 95%. So we want to understand what is the effect of this compartmentalization on the dynamics. And so in order to do that, we need to also consider control scenarios. So the biologically realistic scenario, which we have up here, this model version 1, is that you have a slow exchange of virus between the two compartments, and the CTL move rather slowly from EF to F. Then we can have a control and say, how would the dynamics be if there was really fast exchange between the two compartments, both for virus infected cells and for the CTL? And that'll tell us what this slow movement rate does. And the other control is down here. It's a one compartment model, just basically ODEs mixed. And 2 and 3 are basically identical, because if they mix very fast between the two compartments, that behaves identical to a compartment, to a single compartment model. So we check this out. And so these are simulations of this model of the Gillespie simulation. In the middle is sort of the central graph. This is the average time to reach 95% mutant dominance. And in the purple line here is the first model, the one with the slow migration rates, the compartmental model. And the green line and the brown line are the fast mixing and single compartment models, which are identical. And I would like to say one thing that is sort of important that we do here. We take this model here, the one with the slow migration rates, and we vary the strength of the immune response, and we plot the invasion time as a function of the strength of the immune response. When we use the control models, we make an adjustment. We adjust it such that the amount of virus in these scenarios is identical to the amount of virus in the slow migration model. And the reason is the invasion times are determined by the population size. So if you've got naturally a larger population, it'll take longer to invade. So we don't want that to confound our observations. We want to really see what is the effect of the compartmental structure rather than differences in the amount of virus. So we adjust the strength of the immune response in these control models such that the total population size is identical to the top version of the model here, which is the compartmental model. And so these are the results that we get. And let me just go through these graphs in a bit of detail. So this is the first graph is pretty clear. So the purple line is the two compartment model, and the green line is the mixed model. and this is the single compartment model or the total amount of virus in general. Here we just look at the infected cells, and this is showing what I showed you before. If you've got the 2-compartment model with low migration, F compartment always has high virus load. The EF extrafollicular compartment goes down as the CTL response gets stronger, because that's where the CTL act. If they mix really fast, there's no difference between the 2 compartments, which is also expected. So that's just standard stuff. This is sort of the interesting result that we got from this, and this is looking at the average time to dominance of the escape mutant. And I would like to concentrate on regions where there's a strong immune response here. And you can see that as the immune response becomes strong, in the compartment model with low migration, which is the biologically realistic one, you see a deviation, much longer invasion times compared to the mixed model or the single compartment model. So a standard model, all these models that have been generated, that have been analyzed so far, which are largely a single compartment model, they predict a relatively fast rate of mutant, escape mutant invasion. So you should see the escape mutants after a relatively short period of time. But if you consider this lymphoid tissue compartmental structure with a limited exchange of populations, then the model predicts that there are much longer invasion times. And the reason for this is simply that in the extrafollicular compartment, there's little virus because the CTL controls it so well, and so it's very unlikely to generate the mutant. In the follicular compartment, it's likely to generate the mutant, but it will have difficulty rising because it doesn't have much of an advantage at all. And so this results overall in longer invasion times. And so that's when the CTL response is strong. And this is what for this talk, I want to concentrate on that region, because that's what I think is a very interesting region. So this is that part, this result. And then we also looked at where does the mutant actually get generated when it does invade, in which compartment is it generated. And you can see that mostly it's generated in the extrafollicular compartment, although it's unlikely to generate there. When it does get generated, it there's strong selection pressure that it can rise very fast. If it's generated in the F compartment, then it doesn't have an advantage, may have in fact have a disadvantage, and so it's very unlikely to arise. So the small compartmental size here really limits the time until dominance. If the CTA response is very strong, then they become more comparable, because the amount of virus in the F becomes so low that you never generate a virus here. Then you just have to wait for a very long time until it originates in the follicular compartment and kind of drifts upwards and then moves to the extrafollicular compartment. So this is just looking at where it gets generated. Obviously, if you've got a mixed, if there's high migration rate and a lot of mixing, then they get generated with equal probability in the 2 compartments. So the important result that we think we have here is that with this compartmental model, we predicted it would take much longer for a CTL escape mutant to emerge. And how does that depend on parameters? So typically, the basic reproductive ratio of the virus of HIV is around 8. So this is the simulation that we did. We can do it with lower basic reproductive ratio, higher basic reproductive ratio. So this discrepancy in times seems to get larger with smaller R0. Okay, this is interesting to look at. I'm not sure what to make of it. HIV is around 8, but I mean, there's variation in measurements, and they kind of range from 3 to 20 or something like this. So it's important to see that over all of these R0s, you do see a discrepancy. It's just the discrepancy becomes stronger between the compartmental and non-compartmental models for lower R0.
Okay. Oh, Dominik.
Yeah.
I think we'll be booted out in about 3-4 minutes. Because
seriously? Oh no. Okay. Okay.
I should have warned you at the beginning.
Yeah. Will the Zoom just stop, or?
I think at 1:30 they can see a group milling outside who'll probably.
Okay. Let me just finish it real quick then. Okay. Yeah, I'm almost done. So this is model. The model predicts this discrepancy, and what we see in data is that it actually takes a very long time for escape mutants to emerge in patients, and that has always been a puzzle to people. So you can see that after about 1 year, 60% of patients have not had an immune escape, a CTL escape mutation, and all the non-compartmental models would have predicted that this would have happened by now with a very high probability. So that was always a sort of puzzle. And I think with this compartmental structure, we can explain this. So let me just skip a couple of things. So what we've done so far is escape mutations. I want to just very briefly talk about general mutants that don't necessarily escape CTL or immune responses, but that can just become advantageous due to other things like a faster infection rate. And well, again, we find that for the compartmental model, it takes a lot longer for a mutant to invade. So that's invasion times. We also looked at actually the fixation probability of a mutant, where you put one mutant in and look what is the probability for it to kind of take over, to rise. And we can see some interesting dynamics here, where we can compare to the Moran process prediction, where just a simple calculation, and for low immune responses and for high immune responses, it seems to coincide with that. But for intermediate immune responses in the two compartmental models, you have got weakened selection, where the fixation probability of the advantageous mutant is low. In the one compartment model, you just see it go down as the strength of the immune response is increased. And I can just, this last slide is interesting to explain, because it seems to all depend on fluctuations. So Moran process is a constant population where you have a constant number of cells. But in these CTL model, the predator-prey type model, you can get fluctuations happening. And fluctuations are known to weaken selection. So if you have a one compartmental model and you've got weak CTL, you have small fluctuations in the population. If you've got strong CTL, you've got large fluctuations in the populations. Therefore, it goes down and down and down and down, the fixation probability, because it weakens selection. On the other hand, with the two compartmental model, you also have got small fluctuations for low immune responses. For strong immune responses, you've got large fluctuations in extra-follicular compartment, but that is a very small population. And since there are no CTL in the follicular compartments, there are no fluctuations there either. So this dictates that it's close to Moran process. And if you've got an intermediate strength of immune response, you've kind of got more even fluctuations going on, and then you've got this weakened selection here. And we're still working on that and working that out. So just the conclusions that compartmental structure is basically important for explaining various aspects of virus dynamic and virus evolution. And this brings me to an end, right at 10:30. There you go.